Welcome, everyone. Thanks for coming today. I know it's almost the break, so good job. My name is Pier Andrée. I'm the head of the LMB Flow Cytometry Facility, and today I'm going to talk to you a little bit about some of the things that we set up, the technical considerations in cell sorting specifically. Fan this week discussed kind of flow cytometry on a general level, kind of the analysis, how it generally works. I'll dive in a little bit more into cell sorting specifically, which takes flow into a whole other level. The first slide here, I would just like to point out that there were multiple different technological, let's say, implications in the advancement of cell sorting specifically. And one of those is actually an inkjet printer. And I'll talk about that a little bit later. We also have kind of a Coulter counter here, which was developed by Walter Coulter to enumerate cells. And then we have kind of an image of a modern day sorter at the intercept point with, of course, the addition of lasers so that we could read fluorescence on cells. So I'll just start with talking a little bit about what exactly is flow cytometry. It's a sophisticated cell imaging technology, so very similar in many ways to microscopy, but on a more quantitative level, in which a population of cells are actually hydrodynamically focused into a single cell stream, and they're pulsed with lasers to record fluorescence at different wavelengths. And modern day sorters now can be equipped with up to 9 different lasers, many different types of wavelengths. And of course, not similar to or dissimilar to imaging or typical microscopy, the intercept point of the cell going through the laser is very, very fast. So this avoids any kind of viability issues or kind of photo bleaching effects that you would potentially get with typical microscopy. Fluorescent dyes and probes are obviously used, allowing the analysis of the expression of a variety of different cell markers. We can do proteins on the surface, obviously intracellular expression as well, transgenic products linked to fluorescent reporters. The list goes on and on. So it's quite flexible. And then fluorescent-based cell sorting uses electrostatic deflection to purify populations of interest based on these fluorescent properties. We can also sort cells just based on their forward and side scatter, which Fan touched upon this week. So cell size and kind of a relative cell size and internal complexity or granularity, but you do get better specificity if you're using fluorescence. And it's really the most efficient technology to generate purities of over 99%. So it's really currently the most efficient technology in getting out purifying mixed populations of cells. I like to kind of use the analogy of if microscopy were kind of like the long distance runners, these would be the sprinters. So it's very, very quantitative. And when I say quantitative, we can do actually for certain types of cells, we can do up to 25,000 events per second, which is pretty mind-boggling when you think about it. So I'll just start with an outline for today's talk. I'm going to start a little bit about the history behind the development of cells, of a cell sorter. I'll talk about applications and measurable parameters. How does it work? A little bit about sample preparation tips and guidelines. And then finally, I'm going to highlight 3 LMB flow cytometry projects that I think are really interesting. I think the theme this year has been small, fragile, and very rare. So I think that's very cool. And the 3 projects I'm highlighting will show that. So flow cytometry, cell sorting specifically, was actually developed around 1962 by Mac Fulwiler. And Mac actually was had joined the Los Alamos laboratory in New Mexico, and with him and Marvin Vandila. And their primary interest at the time was actually testing atmospheric nuclear fallout, having atmospheric nuclear fallout testing on food products. And then after the nuclear test ban treaty in 1963 was signed, their kind of mission moved more towards whether they could help the biologists at Los Alamos basically understand a little bit more about cells, and they got interested specifically in the Coulter counter, which I highlighted in my first slide. The Coulter counter was actually developed by Walter Coulter, and this was a device used to enumerate and size particles suspended in electrolytes. And so one of the reasons why they got really interested in developing something a little bit with a bit more specificity is that they noticed that a pathologist that they were working with was incorrectly kind of using the machine. And he was essentially incorrectly identifying what he thought was an immature red blood cell and using the Coulter counter. So Marvin and Mac got together and they thought, actually, isn't there a way to kind of use the Coulter counter, develop it a little bit further so that we can get a bit more specificity in identifying different cell types? And actually, at one point, it was a Fulwiler that read a paper by Richard Sweet, who actually had developed an ink writing oscillograph, as we know now, the inkjet printer. And where, you know, a jet is vibrated, these little drops are broken off into and then charged and then deflected. And he thought, this is a really brilliant idea. Isn't there a way that we could actually incorporate a cell into these drops and then separate them out? So he did that. And this is one of his first sketches. I just love these kinds of things because it's just brilliant, the ingenuity, all of the creativity that goes into developing technology, which is primarily why I was really, really drawn to this field in the first place, and in general, very technical fields. But what's great about it is that he patented this, and it was developed even further by other people. And there is a link very much to the LMB, in fact. After Mac had developed that kind of first prototype, it didn't contain any lasers. And Leonard Herzenberg, who, this is a picture of him while he was a visiting postdoc here at the LMB. He was a geneticist at Stanford, and he immediately recognized that there was this potential to use this technology, and was interested in obviously adding a light source so that he could sort any kind of fluorescently labeled cells for more, again, even more specific fractionation of certain subsets. So he discussed this with Mac, and Mac said, sure, here are the plans for my cell sorter. And Leonard kind of ran with them, added lasers. And then, of course, upon the invention of monoclonal antibody technology by Cesar Milstein and George Kohler in 1975, and there's Cesar here at the LMB, of course, this kind of created the perfect marriage between the field of immunology and flow cytometry. And so now scientists have the ability, or had the ability, to tag antibodies with a fluorescent dye and then label and fractionate these very specific cell populations of interest for, you know, all kinds of things, downstream genetic testing, transplantation, and now it's just so widely used. So a couple of the different applications, immunology is clearly a big one, obviously molecular biology, but pathology, it's even used in marine biology. And what's really great about its use in marine biology, especially these days, is that it basically takes advantage of this autofluorescent property of photosynthetic plankton, and you can characterize abundance and community structure, which I think is incredibly important in the days of climate change. I will now move on to a little bit about what kind of measurable parameters we can sort out populations for. So clearly the first one is cell surface and intracellular antigen expression. So by design, you know, cell sorting in flow cytometry is an a priori bias technique, because obviously we're choosing proteins of interest. That makes it biased, unlike mass spec. So we have to keep that in mind. So protein expression, phosphoproteins, you can do total DNA and RNA content. things like cell cycle analysis, cell kinetics, proliferation, ploidy and aneuploidy, as well as apoptosis. And we do a lot of chromosome sorting actually in the facility. So that's one of the things that we've most recently done. And even, again, cellular pigment. So I, in my couple of past institutes, I even did a bit of plant-based sorting as well. So I think Fan has already touched a little bit upon the basics of flow cytometry and how it works. But all flow cytometers, including cell sorters, fundamentally have the same fluidic setup. So you have a central core through which the sample fluid is actually injected. And then on the kind of outside, it's enclosed by sheath fluid that hydrodynamically focuses that central core. And in a perfect system without any issues, there should be actually no mixing of the sheath fluid on the outer core and then the inner core, which contains your cells of interest. The sample is it's focused into the center by the Bernoulli effect. And this allows the stream or the core stream, the cells, to be interrogated one at a time. So that's why it's known as a single cell, a very high throughput single cell technology. And so that's what makes it so powerful, is that if you can imagine that these cells are going through at 25,000 events per second, it's pretty incredible. And eventually, if you have the machine booked all day, which many people here do, if you need to get through a lot of samples, you can get through tens of millions of cells in a day. So beyond the fluidic part of the system, cell sorting, again, takes this to another level because we're physically separating out populations or cells of interest. And we often, the reason why we use sheath is it's basically a buffered saline, is that it's conductive. And at the top, these are kind of different examples of where the nozzle and the interrogation point of the laser takes place. The very first one, A, this is an example of how an analyzer would work, which Fan covered. So you have your sample inlet, which goes up, and then you have a quartz, normally a quartz flow cell, and then you have an optical interrogation or a focused laser beam there where the cells will pass through, and then it will just go to waste. So we don't sort any cells here. B and C represent two different types of cell sorting technology. They're pretty much the same, but you'll see a couple of fundamental, this is basically the fundamental difference. In B, you have the sample inlet, so it's coming down, it's pressurized, the cells are hydrodynamically focused, and it will hit the interrogation point or the laser beam, and then it will exit out of the nozzle below. And this is known as a quartz cuvette-based system, and C is known as a jet and air, where instead of having the nozzle below the interrogation point, the exit orifice or the nozzle is above the interrogation point. So this creates an advantage in speed mostly because you have lower electronic dead time than the interrogation point here. Some people would argue that actually having a quartz cuvette-based system, as in B, might give you slightly better resolution because this interrogation point here takes in place in mid-air, hence the name jet and air. But they both have advantages and disadvantages. They both basically do the same thing. So some people may prefer the speed of jet and air because they need to get through many, many more cells. If you don't need the speed, then you can easily use a quartz cuvette and it will do the same fundamental job. So how do we create droplets? Basically, we have a piezoelectric transducer at the top that is vibrating at a very high speed, and that creates a the droplets to break off. And it's we set it at a very specific frequency, and that frequency can vary anywhere between 20 and about 90 kilohertz. And then we have this vibration. And then the cells, at the moment the cell is placed into a drop, or the cell of interest more specifically, that cell is actually charged. and then it passes through, I don't have a picture of the plates, but it passes then through in the middle of a series of high voltage plates that are charged either, and then if the particle is charged positive or negatively, it will then be sorted off to the right or left. The cells can have we can sort up to 6 different populations in modern day sorters. So they will have a full positive or negative charge, or a half positive negative charge, or quarter positive negative charge. And that's how we sort cells. A couple of the things that we need to set up when we're QCing and setting up the machines in the morning. One of the most important things is calibrating what's called the drop delay. And this is extremely important in making sure that your purity is accurate, that you have the highest possible purity. And so we know the frequency. We also can have the time taken for the cell to traverse that distance before it breaks off and is sorted. And that time determination is when to charge the stream as the required cell breaks off into a drop. The drop delay is not calculated properly, or your stream is drifting, or the breakoff point is drifting up or down, then clearly the correct cell will not be sorted, and you will have more than likely serious issues with either purity or even having empty drops. In other words, no cell being placed into a drop and sorted. So this is a very important part of ensuring that we get the correct population of interest. One of the downsides of a jet and air sorter specifically is that it takes a little bit longer for the fluidics to settle down. So that's why you'll often see when we come in the morning and we're setting up the machines, we'll really give about an hour for our jet and air sorters to kind of settle down. This is also part of that, because clearly we don't want this breakoff point to be drifting. Having any bubbles in the system will create this problem here. And then obviously not sort your cells appropriately. So quartz cuvette systems are much easier. They're a little bit, because they're more contained, they tend to be a little bit quicker to set up. So that's one of their advantages as well. So a couple of the things that affect droplet formation. The first I've already touched upon is the transducer frequency, which again can range from anywhere from 20 to 90 kilohertz, sometimes actually almost up to 100, depending on the system. If you imagine the vibration of the transducer, the lower the vibration, so once we get down to say 20 kilohertz, the droplets are much bigger. And then the higher it is, clearly the droplets are much smaller. So that translates mostly into speed. The faster we can vibrate on the transducer, the more droplets are broken off and the faster we can actually sort. Another thing that affects the droplet formation is the amplitude. So that's the breakoff point as well. And the nozzle diameter. So cell sorters are equipped with a variety of different nozzle diameters. The most common ones are 70 micron, 100 micron, and maybe 120 or 130 micron. Modern sorters now have kind of a broader, wider variety of nozzle diameters. I'll talk a little bit more about the importance of nozzle diameter vis-a-vis your cell type in the next couple of slides. The sheath pressure, this will also, of course, affect droplet formation. The lower the transducer frequency, typically the lower the pressure. So if we're running frequency around 30 kilohertz, we'd probably have the pressure around something like 20 to 30 psi or pounds per square inch. The higher the sheath pressure, we would be using much smaller nozzles. The highest sheath pressure we normally use is about 60 psi. Some people set their pressures to about 70. Personally, I don't find that there's any major appreciable difference in speed if I just drop it by 10 psi, and I find that the gain in viability is much greater if that's the case. So we usually run it around 60 psi for our smallest nozzle setting. And then finally, the phase of the charging pulse will obviously also affect the droplet formation. A couple of other things that I kind of already slightly touched upon, but one of the things that's very important in a flow cytometry lab is that we keep the temperature quite low. Sometimes people complain and they put sweaters on, but there's a reason for this, and that's because the viscosity of the sheath fluid is temperature dependent. So if we have high temperatures, it can actually affect the stability of the drop formation and, more importantly, the stability of the stream. Debris in the nozzle, this is, I think, probably a fairly obvious thing, but if you bring your samples and they're not properly, prepared, and we get anything stuck in the nozzle, you can imagine that that's going to affect how the droplet is formed and how a cell is placed into a drop, and potentially even clog the machine entirely. So sample preparation is really key. I'll talk about that later. Also a complete or partial blockage of the flow cell, and any air bubbles in the fluidics lines or in the flow cell, which I touched upon earlier. So basically a clean and bubble-free sorter is a happy sorter. Hence why we take our time in the morning to set everything up to ensure that we have kind of a very smooth sorts throughout the day. One of the common questions that we get is how much volume will be generated when we sort the sample. So you can actually approximate this. Some of our newer sorters, our Bigfoot sorters, we actually have a very specific calculation that's available to us. So if you need to discuss this with us, if this is a concern, if you do require lower volumes, definitely just reach out to us. For our ARIA sorter, we don't have a very specific volume generated, but we can use this calculation to approximate that. So if we were using the 70 micron nozzle, we'd probably generate approximately a drop volume of 0.8 nanoliters. This is assuming that a drop is a sphere, so it's not exactly the case, but this is probably a very close approximation. And you can see that as we get, as I mentioned earlier, as we have a larger nozzle and a lower frequency, our drop volumes start to go up quite considerably. So this may be a consideration for those of you that want to have very low volumes generated for downstream sequencing, for example, or even cryo-EM, so purification for cryo-EM samples. So nozzle selection is really critical vis-a-vis the cell type or sample types that you're bringing. Typically, there's a good ratio to kind of go by, and that is typically if the particle size is greater than the nozzle diameter divided by 4, then you're going to have some perturbations. So you would never squeeze through 100 micron cell through, even a 90 micron nozzle. It's not going to work. And so there's a general rule of thumb for a 100 micron nozzle. I typically, recommend 16 to 20 micron max. We can push that up a little bit if the cells are very well prepared. So there's always a bit of leeway. It's not an exact thing. If the cells are less sticky, then we could probably push that up to even, 25 or even 28 microns. But then at that point, we'd probably prefer to go and use a bigger nozzle. So one of the other considerations that you want to think about is that it's not just kind of this perfect ratio between the cell size and the nozzle size. It also, you also have to consider the viability or the sensitivity of the cells being sorted. So as I mentioned earlier, there are, when you go up with very larger nozzles, you'll have a much lower pressure. And so that might be more advantageous if the cell type that you're trying to isolate is extremely sensitive. And so even though it might be small, it actually sometimes is better if we actually use a larger nozzle and a lower pressure. So we'll change up gears now a little bit, and I'm going to talk about sort modes. There are 3 different distinct types of sorting modes. There's a little bit of play in between these as well, but this is kind of a general overview. So the first is where, if in your mind, you really want to get the highest possible number of recovered cells, and that's the primary concern. And in this case, each target particle is sorted, whether or not there is a contaminant particle that may be brought along with it. So here you have recovery, very high recovery typically, but at the expense of a little bit at the expense of purity. The second is probably the most common situation, and this is where people are very, very, keen on making sure that it's very high purity, and they don't care so much about, recovery. Again, that we can tweak this a little bit, some of the sort modes where we have kind of a happy medium where we still get very high purity, but we kind of maximize a bit more of the yield. And that's where the target particles are sorted if no coincident particle is detected, and if no known contaminant particle is included in the droplet being sorted. And then the final scenario is typically done for single cell cloning experiments, where you want the maximum achievable purity regardless of the cost and yield. And that is where we sort a particle, contaminant, or target in a 3-drop envelope surrounding the target particle. It causes the sort to be aborted. So it's very, very stringent. I have a slide talking, kind of showing a little bit how this works. So there are what we call sort envelopes. So there's, give or take, depends on the system, but 5 different ways to sort a target. So 1 drop is deflected only if it's centered, only if the particle is centered in the drop. 1 drop wherever it's located in the drop, so it doesn't matter if it's on the edge or anywhere. You can also do 1 or 2 drops deflected, 1 if it's centered, and 2 if it's close enough to the edge that it might drift in the next drop. Always 2 drops deflected, or always 3 drops deflected. And all of these sort envelopes, again, really depend on the aims of your experiment and the type of cell. And I'll show you in the next couple slides how this works. So as I mentioned earlier, you have yield, purity, and then single cell mode, which is the most stringent. So if we look at the masking, so if you look over here on the example side, you have the drops that are divided into like a 32 drop window or envelope or a mask. So in the first case with yield, you have your yield mask to the maximum. So you can see that no matter where that cell, the target particle in gray is located, even if there is a non-target particle in red close to it, or even contained within the same drop, we'll actually sort all of these particles. In the purity mode, is quite different. The purity mask is set to 32. So that means if you look at the first scenario on the left, the non-target particle that's within that masking of the previous drop, that means that it will not sort the target particle because that non-target particle is too close that it may drift into the drop of the target particle. And the same goes with on the other side here. So you can see in the middle case, the non-target particle is outside that purity masking. So that means that will be actually sorted. And then finally, in the single cell mode, it will only sort the cell if it's actually, if the target particle is placed in the center of the drop. So if it's not in the center, it won't sort it. If there's any kind of contaminant particle within that 32 purity masking, it won't sort it. It definitely won't sort something if there are 2 cells in a drop as well. One that's the non-target particle and one that's the target particle. So here's an example of how sort envelopes could work to your advantage or disadvantage depending on the sample type. So here I actually have an example of a bacterial filament that, depending on your drop envelope and depending on where the break-off point is calculated, you can see that in this case, because it's a filamentous particle and it's quite long, it can actually be present in multiple drops as it's being sorted. So if we actually use a 1-drop enriched envelope, you can see that part of that filamentous bacteria will actually not be sorted correctly. If we extend the drops to sorting 3, we can actually take advantage of that and then have the full filamentous bacteria sorted properly. So this is why it's really important to give us as much information as possible prior to kind of thinking about your experiments. A question I often ask people is, what's the morphology of what you're sorting? Is it, especially when it comes to bacteria, is it spherical? Is it filamentous? How does, what does it look like? And this is part of the reason why, because we can adapt the settings to the sorter kind of to maximize the purity and the recovery of your cells being sorted. So finally, in terms of recovering your cells, you should consider that not all of the cells sorted into a tube will be recovered after you spin them down. One of the main issues here that we encounter, there's a couple of things. The first is making sure that when you bring your collection tubes of interest, that you bring them with some kind of media inside. And also we want to maximize the centering of the sort of the side streams into the center of the collection tubes. And that's so that we maximize the chances of those drops hitting the media and not the sides of the tube. If you imagine a cell coming down at a fairly fast velocity and it's hitting the side of a plastic tube, you're not going to get very good viability potentially. So what we need to consider is how many of the cells of the ones we want are actually deflected as a measure of sorting efficiency. So that's the percent sorted of the desired population. Normally what I tell people is if you have a high enough number of cells, you should always spin them down after you've sorted them and then resuspend. You should get a pellet, you should see a pellet. It will also give you a number on the side of the tube, letting you know approximately how many particles or cells are in your tube. So I tend to encourage people to, if you have enough, sometimes this isn't possible if you're only sorting a couple hundred cells, but if you have enough, take an aliquot and measure how many cells are actually in there. So just verify that. Because it's good for us to know that the recovery is quite high. And then one of the ways that we check purity is once we're done sorting the population of interest, we will, if again there are enough cells, we can take an aliquot from that population and then rerun them through the machine to ensure that the population of interest was in fact sorted and that it's validated as very pure. And when we talk about purity, and I haven't mentioned this yet, but I'm talking about anything over 95%. I would say anything under that is probably not great. So it really should be over 95 to upwards of 99% purity. One of the things that we do when we're setting up the machines is we're ensuring that obviously our stream is stable, making sure that our charge phase and all of the things that we've set up to have accurate sorting, particularly in the generation of the side streams, that they're stable. But one thing that we will notice right away is if your sample is not properly prepared or if it's too concentrated or too sticky, the minute you put it on, it will actually cause the side streams to fan and become unstable because you're basically trying to push either your cells are too big for the nozzle type, or they're too concentrated, or they're simply too sticky. So basically creating a non-Poisson distribution as the cells are going through the core stream. So one of those things that we do to try to mitigate that is to make sure, if your cells are rapidly dying, maybe add a little bit of DNase to the media. If there's sticky cells, make sure that you're adding maybe a little bit of EDTA. dial down the concentration, filter your cells. All of these things will mitigate those problems. And again, getting back to the more you know about your cells in advance, the morphology, the expected size, the better we can kind of guide you to the correct, machine, knowing what settings we're going to use to maximize your results downstream. All right, so a little bit about sample preparation guidelines. So successful flow cytometry and specifically successful cell sorting really requires that you pay a lot more attention to sample prep than is probably typically done when you're just analyzing your cells, although you should also make sure that your cells don't clog the analyzers, so you'll get unhappy people who won't want to run their cells after you because you've been clogging the machine. But really the challenge lies in balancing your fractionation of your population of interest in kind of a timely way to preserve viability, functionality, especially functionality, and expansion and culture for downstream assays. And another prerequisite is to have a good single cell suspension so that, again, you're ensuring that when the cells are flowing through the core stream, that we're also minimizing any kind of doublets. So those are two or more cells that are stuck together due to kind of inappropriate sample prep. So I often see people bringing their samples in RPMI or DMEM, and they're actually not the best candidates for sort buffers or collection buffers. So one of the reasons is that they are a different buffer than the cytometer's sheath buffer, which means basically you're looking at a bicarbonate versus phosphate-based buffer. And by design, they also require 5% CO2 to maintain physiological pH. So if we're sorting cells for a few hours, that's obviously going to become a problem. And they also contain divalent cations, which also can cause a problem, and also including phenol, which is very autofluorescent. So we want to reduce that as much as possible. So if a bicarbonate media is used, you should be wary and use either calcium or magnesium-free formulas without phenol to mitigate any undesirable effects of the cation side effects for sorting. So what happens in that case is that it makes the cells quite sticky. So again, causing issues with downstream separation. You can add 1 to 2 millimolars EDTA and 25 millimolars of HEPEs. So keep in mind that HEPEs buffered bicarbonate media is light sensitive. So just keep it outside of, protect it from light. All right, so a few more tips and tricks. So first, if you're using trypsins, make sure you inactivate it with soybean inhibitor or use Accutase. Cells grown in any media and serum should be rinsed. They should be rinsed at least twice with PBS prior to dissociation. Make sure you test your dissociation, especially with non-enzymatic buffers relying on any chelating agents. Always use the highest quality reagents. I think that probably goes without saying. And then finally, make sure that you remember that collagenase is dependent on calcium for activation. A little bit now about the appropriate types of collection tubes and buffers for your sample. So the best practice is to actually pre-coat your collection tubes with serum. That, again, tends to kind of mitigate any effects with the cells hitting if they hit the side of the tube just by chance. Then if they're pre-coated, it will help them slide down into the media. Use the same sample buffer for the collection tubes with 2 to 10% serum in your collection tube if desired. The process of sorting cells can actually be, quite harsh. You try to mitigate that, but if the cells are kind of landing in a kind of a happy protein-rich environment, it will kind of help them along. Again, any media added to the wells should be HEPEs buffered and conditioned beforehand. I already mentioned not to use any unbuffered bicarbonate media. If you are sorting very rare populations, we always recommend smaller collection tubes. So we can either sort into multi-well plates of various different sizes, 384, 96 well, basically whatever it is, we can more or less optimize it and sort into it, or even Eppendorf's or micro PCR tubes for really, really rare subsets. So we can really refine that even further to very, very small tubes. And that will improve your recovery quite a bit. And we do recommend polypropylene tubes over the polystyrene fax tubes. And it's mostly because the cells adhere less to the tube wall in the polypropylene tube. Basically, polystyrene tubes can add to a buildup of charge in the deflected drops on the surface. And sometimes if you use polystyrene, this is why when we have if someone brings polystyrene and they really don't have polypropylene, as the tube is filling up and it's getting closer to the top surface, if you imagine you have several different tubes which are not very far from one another of different populations, that buildup in charge can actually cause jumping drops to go in and then kind of contaminate. So we tend to not sort all the way to the top. We'll stop at a certain point to give a bit of buffer there so that we mitigate that. Also, one of our sorters, the Bigfoot sorter, has what's called a polarity flipping. So we can set that up to also mitigate any additive effects of charge inside the sorted cell tube. So essentially what the polarity flipping will do is we can set it to every certain amount of volume of cells passed, it will actually switch the charge. So if it sorts positive, if it applies a positive charge, then it will start applying a negative charge. So that will have an effect of a net neutral charge of the drops in the collection tube. And that's quite helpful actually for neuron sorting. All right, so I'm going to talk about 3 different projects that I think are really, really interesting. And they were, quite challenging in their own right for some of the scientists here that wanted to sort these cell types. The first is Miranda Robbins in the Zlatic lab. And she's actually looking at molecular changes required for memory formation in Drosophila. And her project involves determining whether different dopaminergic neurons and their postsynaptic cells involved in reward learning have different naive transcriptomes prior to learning. And that will elucidate any that kind of understanding of the complexity of molecular signaling that can occur in the circuit. For example, how many transmitters or receptors, or even sensitivity to calcium. And Dr. Julie Simpson, who is in Marta's lab, she's a visiting scientist from UC Santa Barbara. She's looking at whether motor neurons required for grooming have different molecular properties. So this is a fax plot of Miranda's samples. She's labeled her neurons with amnion green. To give you context here, this is actually over 1 million cells, and we have probably about 10 cells that are positive for neon green. So it's very, very challenging for 2 reasons. One, they're extremely rare, and 2, they're fragile. So she's sending these samples off for sequencing. We should have the results very soon. But to give you context, after running the samples, the total number of cells I would get would be about 50 if I was on a good day. So I get typically 2 duplicates of 50 cells. So, and we sort it into micro PCR tubes to make sure that we maximize the recovery as much as possible. So I'm really looking forward to seeing the results of this. The second really interesting project that came into the facility is Chris Kuo from the Baum lab. And he's looking at cell division in archaea. And Sulfobulus acidodelicaris is a thermoacidophilic archaea, and it's a facultative autotroph, and it's found in thermal soils and hot springs. And the goal of one of Chris's projects is to isolate cells at different substages during the cell division and pinpoint at which substage the transcription of cell division genes are turned off. And qPCR analysis on the sorted cells actually show that the transcript abundance of the division genes peak when the division ring is constricting, and then it's rapidly turned off upon completion of division. So this is, I think, is a very cool project. I love the fact that we're sorting all different kinds of things. It's one of the things that probably attracted me most to the LMB. So I'd never sorted archaea or even worked with archaea before. So here they are. We have them sorted. Basically, he's using Huxt, and he's using another dye as well, like the 448, and then we're sorting the various different division cycles here. So also very challenging, but probably from a different angle. The archaea here are about 1 micron, so they're quite small, but definitely, certainly easy enough that they're still well within the range of resolution limits of the machine. I think one of the things we weren't sure about initially when we kind of started sorting these is whether they would survive, but I think they're so hardy that I think they were happy to go through and there was no problems at all. So it was very cool. And then finally, I just wanted to highlight one very interesting project by Michelle Coe in the Mackenzie lab. And she's actually looking at the interaction between enteric neurons and innate lymphoid type 2 cells. And she's seeking to understand how the enteric nervous system and immune cells interact to regulate defense response against helminths or parasitic worms. In the intestine, there are hundreds of thousands of neurons and glia that coordinate all the necessary functions of digestion, nutrient absorption, defense, all these things. But she's very focused on a specific population of enteric neurons that express molecules known to regulate ILC2s. And of course, it would be very difficult to study one subset of neurons in a very heterogeneous sample. So that's where cell sorting comes to work in her project. And she's labeled these neurons in a transgenic mouse strain where they express TDRFP. And this has never been done before, actually. So, and then she is culturing them to perform downstream transcriptomics and also for in vitro assays. So again, one of the things that's quite challenging about Michelle's project is there's a couple different things. One is actually just dissociating these and getting them into a single cell suspension without causing any kind of perturbation and preserving viability. And then the other challenge is that, as you know, the morphology of neurons is such that they're not probably the easiest thing to sort. They're quite spindly. And so we weren't sure whether they would survive. And so we played with a couple of different settings. We also played a lot with the polarity flipping on the Bigfoot to again ensure that the neurons we'd have a net negative charge in the collection tube so that they weren't so activated after sorting. And this is a beautiful picture of them growing up day 19 post-sort. So it's very happy to see that. I love to see happy cells post-sort. It's one of the things that we strive for. So, all right, so just to end up my last couple of slides, finish up, I'm going to talk a little bit about the future developments in the field. One of the things that I think in my decades-long career in flow cytometry that's been so fascinating is actually seeing the changes in the field, particularly in this kind of very interdisciplinary or cross-disciplinary overlap with other types of technologies. So, and that can be anything from mass cytometry, where you incorporate elements of mass spec with cytometry. But one of the most exciting, I think, parts of the field right now, or changes in the field, is actually overlapping imaging with sorting, and imaging specifically with flow cytometry. In 2022, this brilliant paper was released where they essentially created a machine where it's image-enabled cell sorting at very high speeds. When I say high speeds, here it's about 10 to about 15,000 events per second, which is quite amazing. And three crucial components of the development of this image cell sorter were that they used fluorescent imaging using radio frequency tagged emission. They incorporated this into a traditional cuvette-based droplet sorter. So this is from BD Biosciences. They use the BD Melody sorter and very low latency signal processing and sorting electronics, which is quite amazing if you think about having an image and sorting based on an image and getting subcellular localization. So this is not traditional with flow cytometry. You would never get any kind of subcellular localization. You would just get generalized fluorescence. So, and they made this into a machine where we could actually fractionate out cells of interest. So the field is definitely changing. It's changing very rapidly. And I think this is the future of flow cytometry, most certainly. People have been working around the world to get this going for, I would say, well over a decade now, but we're getting there. This is a little bit, this is an image taken from the paper. So to enable blur-free imaging, because that would be one of the things that you think right away would be a huge challenge, at a very high nominal flow speed of about 1.1 meters per second. So they use the FIRE approach. As I mentioned, it's a radio frequency approach to produce an array of 104 laser spots across 60 microns within the core stream. And they're each modulated at a unique radio frequency. So there is an acoustic optic deflector, and it splits the single laser beam. In this case, the laser is a 488 nanometer laser. So there's only one imaging laser, and that's 48 nanometers. So it splits it into an array of beamlets, each having a different optical frequency and angle. And then a second acoustic optic deflector tunes that optical frequency of the reference beam. And that is then overlapped with the array of beamlets. And the overlapping beams intersect at a flow cell of a traditional cuvette sorter. And then what they did was, for proof of concept, they sorted and they stained for the nucleus, the Golgi. You can see here on the left where you can clearly see some separation of all the different, even up to P bodies and the nucleolus, et cetera, ER. And they sorted and did CRISPR screening. So it's a really exciting time in the field. You can imagine this kind of opens up a whole new types of applications that we can then explore. The first one that comes to mind is cell painting or morphological profiling. So staining things like mitochondria or nucleus or ER, all of these things, and sorting based on the morphology as opposed to any kind of biased protein expression. High throughput functional genomic screening is huge. The quantitative analysis of any internalized bacteria or parasites, the evaluation of asymmetric cell division, and analysis of even cell interactions and immune synapse. So I think this is, again, a really exciting development. Some of the current limitations of image-enabled cell sorting right now, there's, as I mentioned, only one laser and 4 fluorescent detectors, plus a bright field, dark field for imaging. So here in this case, they say 4 fluorescent detectors. In reality, it's really only one. So it's really good to always, when a company develops something, you want to sell it, do your homework, because I think in reality, it's maybe only 1, maybe 2 fluorescent detectors that we can use on the imaging modality. It also has other non-imaging lasers. I think it has a violet and a red laser that have a few detectors. That if you wanted to actually stain for and get imaging, you kind of only have maybe 1 or 2 detectors off of that would have fluorophores excited at 48. Realistically, I think initially when they released the machine, they were advertising at approximately 1 micron resolution. From what I hear from colleagues, it's more like 3 microns resolution. So we're not really, getting, super resolution cytometry here. Certain applications will require cell fixation, so that kind of negates any kind of purification of anything live. And I would say the last one, again, it's going to require a lot of in-depth knowledge of both imaging and cytometry modalities. So this is, I think, in my opinion, that can be, very exciting, because if you're very happy to learn new techniques and you want to kind of stay technologically kind of state of the art, it's very exciting. But for some people who don't have an imaging background and who are potentially running facilities, it requires a whole new level of knowledge and kind of gaining that knowledge to make sure that you're keeping up with the field changes. The next frontier really is super resolution cytometry and cell sorting. This is absolutely going to happen, I believe, in my lifetime. The cell sorting part will be far more challenging, but I think we will most certainly see super resolution analysis cytometry happening within the next, I would gather, 5 to 10 years. The cell sorting decisions are very rapid. So to have super resolution and blur-free imaging is going to be much, much more challenging, but I am very confident I will see this in my lifetime. So I think the field right now is absolutely exciting, and look out for what's coming, because I think it's going to be very fun. All right, so I'll just end by highlighting the people in our facility, our wonderful staff. So Fan, who's our deputy, and Yi Chen, who is one of our specialists. We're really happy to help make the most of your experiments, and we really welcome working with challenging samples. I think that's one of the things, again, I mentioned it before, that I love working at the LMB for this reason, because you're always bringing very challenging samples from all different types of research applications and backgrounds. So it's very fun. We're always learning, so that's great. We all have a depth and breadth of expertise across various technologies and applications. So I think that's another thing. We all have our strengths, and so please come and talk to us. And if your experiments are part of a larger pipeline across facilities, for example, you're purifying something for downstream mass spec or any kind of imaging, particularly cryo-EM, really think about the power of cross-facility communication to facilitate and accelerate the success of any kind of downstream experiments. It's really helpful for us to understand why you're sorting and what you're doing with the cells after, because again, based on all the things that I discussed today, we will tweak things to maximize that success. And finally, please make sure you formally acknowledge the facility in the acknowledgements after you've used any part of the facility. Please make sure that's included in the acknowledgements section of your publications. And that's it. If there's any questions, I'm happy to answer them. Thank you.